everyone. Today we are going to study basic concepts of DNA, RNA as well as genes from the subject elements of biotechnology. First of all, let's look at the structures of gene. Breakthrough to discovery. This is Watson and Crick, the two scientists who described the structure of DNA in the year 1953. They had the double helical structure. They decided the double helical structure of the DNA and they got Nobel Prize for this. This is the structure of DNA. DNA is a double stranded structure. It has a ribose sugar, that is deoxyribose. Then it has a phosphate group and it has a sugar phosphate backbone, and there are nucleotides. A purine in a DNA always links with a pyrimidine with the maintenance structure of the DNA. The purine and a pyrimidine are linked together by the hydrogen bonds, that is adenine A binds to thymine T with two hydrogen bonds as well but guanine, that is G, and cytosine, that is C, is binded to 3 hydrogen bond. This is the structure of DNA. This is the diameter of DNA is 20 angstrom. If we see, the one turn of DNA is 34 angstrom and there are 10 bases in one turn. So distance between two nucleotides is 3.4 angstrom. The chemical structure of DNA is like this. It has a sugar phosphate backbone and then there are the base pairs through which they are linked. The double strands are linked together with the hydrogen bonds. This is the structure when we look from outside, but this is the structure. If we remove the turns, we can see it like this. Now the nucleoside and nucleotide are the fundamental building blocks of DNA. As we can see the structures here, it has a deoxyribose, that is one of the oxygen is missing, and then it is binded together by the base, that is either adenine, pyrimidine or purine, that is adenine, thymine, guanine or uracil, and it is also has a phosphate group, that is a phosphoric acid linked together. This is how it looks. This is known as a nucleotide. When it is removed, it becomes a nucleoside, that is removal of H2, then it is a nucleotide. This is a phosphodiester bond, that is between the ribose sugar, deoxyribose sugar and the phosphate group, and between the base, that is pyrimidine or purine, and deoxyribose we have the glycosidic bond. This is the 3D structure of DNA. DNA looks like this. Nucleotides have 3 characteristics, components. One is a nitrogenous group, base. Second is a pentose group, a phosphate, and the molecules with the phosphate group is known as a nucleoside. The nitrogenous bases are derived of the 2 parent components, that is pyrimidine and purine. This is the structure of a pyrimidine, that is one of the base, and this is the structure of a purine that has 2 rings with it. The purines are adenine and guanine. This is the structure of adenine and this is the structure of guanine. While the pyrimidines, there are 3, that is cytosine, uracil and thymine. These are the structures of all the 3. This is the nucleotide. As you can see, this is the deoxyribose sugar and this is the adenine and this is the phosphate group that is attached together. This makes the whole one base pair of the DNA. So the adenine group, and now the chemical and physical properties of DNA. Let's look at the chemical and physical properties of DNA. Physical properties of DNA. First of all, DNA stores the genetic material information. Many lines of evidence show that DNA bears the genetic information. In particular, the Avery McClure-McCarthy experiment showed that the DNA isolated from one bacteria strain can enter and transform the cells of another strains, endowing it with some of the inheritable characteristics of the donor. The Hershey-Chase experiment showed that the DNA of the bacterial virus but not of its protein carries the genetic material for replication of the virus in the host cell. DNA sequence adopts unusual structure. A number of sequences dependent on the structure variation have been detected with the larger chromosome that might affect the function and metabolism of the DNA segments in their intermediate vicinity. A rather common type of DNA sequence is the palindrome, that is, this is known as a palindrome. A palindrome is a word, phrase or sentence that is spelled identical reading either forward or backward. For examples, rotator means you read it from here or from backward, both are the same, and nurses run, so if you read it from here also it is the same. The term is applied to the regions of DNA with inverted repeat of base sequence having two-fold symmetry over the two strands of DNA. This is a palindrome, means if you read it forward as well as if you read it backward, it is the same. It is known as a palindrome. Such sequences are self-complementary with each strand and therefore have the potential to form hairpin or cruciform, that is the cross-shaped structures. When the inverted repeats occur within each individual strands of DNA, the sequence is known as a mirror repeat. Mirror repeats do not have complementary sequences within the same strands and cannot form hairpin or cruciform structures. There are three types of DNA, that is the A DNA, B DNA and Z DNA. A DNA is formed by the RNA. This is the A DNA, it has 28 Armstrong in one turn. B DNA, the standard double helix DNA under the physiological condition, and the Z form, that is the laboratory anomaly. It is left-handed, it requires alternate G and C nucleotides and high salt and charge neutralization. Now the chemical properties of DNA. Absorption, the base in DNA absorbs ultraviolet at the wavelength of 260 nanometer. This absorption can be monitored using a spectrophotometer. This is one method used to figure the concentration of DNA in a solution, means how much DNA is present in a solution can be found because of this chemical property of the DNA. The more DNA present, the higher the absorption. Density. Density can be measured by the CSCL density ultracentrifugation. It can be used to estimate G plus C content of the DNA. G and C bases are more dense than the AT bases pair. Density study shows that the existence of the satellite DNA. Now denaturation. What is denaturation? DNA is considered denatured when the double-stranded DNA molecule is converted into two single-stranded molecules. We know that both the two single-stranded molecules are bound to each other with the hydrogen bonds. As thermal energy increases, the hydrogen bonds, as we all know, it's a weak bond, so it breaks off, and the hydrogen bonds breaking between the molecules increases. So the double-stranded DNA becomes single-stranded DNA. This is known as the denaturation of DNA. G and C base pairs are held together by 3 hydrogen bonds, while A and T are held together by 2 hydrogen bonds. So the more the amount of G and C, it is more difficult to break the DNA strands. But if we have more amount of A and T, it can be broken down easily. Now DNA again forms hybrid. The ability of two complementary DNA strands to pair with one another can be used to detect the similar DNA sequences in two different species or within the genome of a single species. Hybridization techniques can be varied to detect a specific DNA or rather than RNA, because RNAs are small, so it can be detected easily. Applications of this technology make it possible for the identification of the individual on the basis of a single hair. Means even if a single hair is left in a crime scene, we can detect them by detecting the DNA. Means in the season of crime, in the scene of crime, or a prediction of the onset of a disease of a decade before the symptoms appear. This is how the DNA hybridization occurs. First of all, we denature the whole DNA, so it becomes single-stranded DNA. Means one sample of one species DNA that is denatured and the second sample of the second species of the DNA that is denatured, and both the solutions are mixed, and then the favorable condition, means as denaturation can be done by heating, so then the favorable temperature is given, so the hybridization occurs. Means wherever there are same complementary nucleotides or base pairs, the hybridization occurs. This is how DNA hybridization is done. Now nucleotides and nucleic acids undergo non-enzymatic transformation. Purines and pyrimidines, as we know, both of them are present in DNA. Along with the nucleotides of which they are a part, undergo a number of spontaneous alteration in the covalent structures. The rate of these reactions is generally very slow, but they are physiologically significant because of the very low tolerance of the alteration in its genetic information. Alteration in DNA structures that can produce permanent change in the genetic information encoded in the protein are known as mutation. Because of mutation, we can have various diseases such as cancer also. So this suggests an intimate link between the accumulation of the mutation in an individual organism and the process of aging as well as carcinogenesis. Now, hydrophobicity of a solvent. Hydrophobic substance will allow the bases in the DNA to dissolve, whereas the hydrophilic substances will keep the bases of DNA stuck up to one another in the orientation that most favors the hydrogen bonding between the DNA strands. Now, pH. Acidic pH causes a breakage of phosphodiester bonds between the nucleotides and the breakage of the N-glycosidic bond in between the sugar and the purine bases. Alkali, that is above the pH 7, means around pH 11.3, all hydrogen bonds are disrupted and the DNA is totally denatured, means alkali or acidic, both can denature the DNA or break the hydrogen bonds. Salt will stabilize the DNA double helix. These are the, now we will study RNA. There are three major classes of RNA, that is the messenger RNA denoted as mRNA, which codes for protein, ribosomal RNA, that is rRNA for translation, transfer RNA, that is tRNA for translation. It is present in all the organism. This is the difference between DNA and RNA. It has ribose sugar, it has deoxyribose. This one has uracil, DNA has thymine. Presence of hydroxyl group at the 2 position, lacks the hydroxyl group at 2 position, because of which it has deoxyribose sugar. RNA is usually single stranded, DNA is usually double stranded. mRNA transcripts the structure of genes, encoded all the information necessary for the synthesis of the polypeptide of the protein. Intermediate carrier of the genetic information delivers genetic information of the cytoplasm. This is how the basic amino acids are coded. There are triplet codon, this is known, means 3 codes, means 3 nucleotides base or code for 1 amino acid, and that is how the protein are formed. These are the structures of the amino acid. Now, tRNA. RNA molecules 70 to 100 nucleotides long. The secondary structure of tRNA resembles the D loop, anticodon loop and a T loop and the acceptor stem. As you can see, this is anticodon, acceptor stem. Carry the correct amino acids to their position along with the RNA template to be added to growing up polypeptide chain. rRNA, the central component of ribosome, it is present in ribosome, that is the ribosomal RNA. It is a factory of protein synthesis composed of the ribosomal RNA and ribosomal protein. rRNA provides a mechanism for decoding the mRNA into amino acids. It decodes the triplet codon, means the triplet nucleotides can code by the help of this RNA. rRNA interact with the tRNA during the translation and providing the peptidal transferase activity. Now, biological roles of RNA. RNA is a genetic material for some viruses. RNA functions as an intermediate, that is mRNA, between the gene and the protein synthesis machinery, that is the central dogma. RNA functions as an adapter between the codons in the mRNA and amino acids. RNA serves as a regulatory molecule which through the sequence complementary binds and interferes with the translation of the certain mRNAs, and some RNAs, enzymes that catalyze the essential reactions is in the cell, that is RNase, P, ribozyme, large rRNA, cell splicing introns, etc. Now, organization of DNA. The DNA is very complex or compact. It is compressed, means this is the DNA. It becomes histones. It is bound around the histone proteins, and then it is coiled and coiled to form the chromosome. Means it is very compressed and compact structure, and these chromosomes are present in the nucleus of the cell. There are 23 pairs of chromosome in the cell. Now, chromosome is the double-stranded DNA molecule packed by the histone and scaffold protein. This is how it is all compressed into forming the chromosomes. Now, what is genome and gene? Genome, the totality of the genetic information of an organism encoded in the gene. For some virus it is RNA. Gene is the basic unit of inheritance. This segment is very long strand of DNA with specific instructions for the production of specific protein. Genes are located on a chromosome, on its place or on locus. Now Chargaff's rules of equivalence. Chargaff rule states that the DNA for any cell or organism should have 1:1 ratio of base pair, that is pyrimidine and purine should be in the ratio of 1:1. More specifically, that the amount of guanine is equal to cytosine and the amount of adenine is equal to thymine, that is A should be equal to T and C should be equal to G. They were discovered by the Austrian scientist Erwin Chargaff. Deoxyribonucleic acid, that is DNA, is the genetic material found in the chromosome of all animals and plants. It is made up of only for 4 types of organic nitrogenous bases, that is adenine, guanine, thymine and cytosine. Of these, A and G are the purines and T and C are the pyrimides, as we have seen earlier. Thank you.